Good morning, everyone, and thank you for being here today. I am pleased to welcome you to this presentation. Today, I will be talking about the generation of in-house working standard for QC analysis. We'll explore how we develop and utilize these standards to ensure quality control in our processes. Over the next few minutes, I'll walk you through the key steps involved and the significance of this work. I'm excited to share this with you, so let's get started. Welcome to this part of the presentation, where we focus on the importance of reference materials in pharmaceutical quality control laboratories. These labs rely heavily on reference materials to ensure that our analytical testing is both accurate and reliable. Now, when we talk about reference standards, there are two main types we should keep in mind: primary reference standards and secondary, or working standards. Primary standards are the most authoritative and are used as the gold standard for calibration. Secondary or working standards, on the other hand, are prepared in-house. They are qualified against an official pharmacopoeial reference standard, which means we can use them routinely in the lab. This setup helps us maintain consistency without constantly relying on the more expensive official standards. The key benefit here is traceability. Working standards are linked back to those official standards, ensuring our results are trustworthy. Plus, they're more practical for everyday testing, helping us save costs while maintaining quality. So, in essence, working standards are a vital tool for balancing accuracy, cost efficiency, and traceability within our quality control processes. When it comes to generating working standards, there are several key regulatory guidelines that we need to follow. These regulations are in place to ensure the quality, consistency, and safety of our products. First, they require traceability to a primary reference standard, which means we must be able to trace our standards back to an official, recognized source. This helps verify the authenticity and accuracy of our standards. Next, qualification documentation is essential. This involves thoroughly documenting that our standards and procedures meet the necessary quality criteria. It's about proving that everything is qualified and suitable for its intended use. Controlled storage and labeling are also critical. Proper storage conditions and clear labeling help maintain the integrity of our standards over time and prevent mix-ups. Additionally, periodic requalification is necessary to ensure standards remain valid and reliable throughout their life cycle. Regular reevaluation helps maintain consistent quality. Now, these regulatory requirements are outlined by several key guidelines. These include USB General Chapters, ICH Guidelines, WHO QC Laboratory Guidelines, FDA 21 CFR Part 211, EU GMP, and ICH Q7 for active pharmaceutical ingredients. All of these frameworks work together to create a comprehensive approach to quality control. Understanding and adhering to these regulations is fundamental to maintaining compliance and ensuring our products meet the highest standards of quality and safety. A reference standard, also known as a primary standard, is a crucial element in our analytical processes. It's a material that is highly pure, meaning it contains very minimal impurities, ensuring accuracy in our measurements. It is fully characterized, so we know its properties in detail, which helps us establish reliable benchmarks. Additionally, it is certified by recognized pharmacopoeial authorities, giving us confidence in its quality and traceability. Some common examples of reference standards include those from USB, BP, EP, and IP. These standards are essential tools for various purposes. They're used for method validation, to confirm that our analytical techniques are accurate and reliable. They help in instrument calibration, ensuring our equipment provides precise readings. They are also critical for qualifying working standards, verifying that our day-to-day standards are consistent. Lastly, they support analytical method verification, confirming that our methods perform as expected. Overall, reference standards are the backbone of quality control in pharmaceuticals, ensuring our results are trustworthy and compliant with regulatory expectations. This ties directly into the overall theme of maintaining rigorous quality and accuracy in our analytical work. Let's take a closer look at what a working standard, or WS, actually is. A WS is a secondary standard that has been qualified against a primary reference standard. Essentially, it acts as a reliable, consistent benchmark for routine quality control testing. Now, some key characteristics of a WS include that it is prepared in-house, which gives us control over its quality. It also typically has a slightly lower purity compared to the primary reference standard. This is expected and accounted for during qualification. Importantly, before we use it in any testing, it must be fully qualified to ensure accuracy. The main purpose of a WS is for routine assays and impurity testing, meaning it helps us verify the quality of our products efficiently. As for its applications, a WS is used in assay determination to measure active ingredients, in dissolution testing to evaluate how a drug releases, in content uniformity to ensure consistency across doses, and in impurity profiling to identify and quantify contaminants. Overall, understanding the role and proper qualification of a working standard is crucial for maintaining the integrity of our quality control processes, ensuring our products meet the necessary standards consistently. Let's talk about the need for an in-house working standard. First, creating your own working standards helps to reduce the cost of reference standards, which can be quite expensive when purchased commercially. By generating your own, you also prevent the frequent opening of these costly reference standards, helping to preserve their integrity and extend their usability. Ensuring continuous laboratory operations is another key reason. When you have your own working standards, you don't have to wait for external supplies, which can cause delays. It also allows you to maintain traceability to official pharmacopoeia, ensuring your testing remains compliant with regulatory standards. Furthermore, having a reliable material on hand supports routine quality control analysis, whether it's for assay testing, dissolution, content uniformity, or stability testing. These are all critical areas where consistent, accurate standards are essential for producing safe and effective pharmaceutical products. Overall, developing in-house working standards is a strategic move to improve efficiency, cost-effectiveness, and quality assurance in your lab processes. When selecting a working standard, it's crucial that the candidate material meets some strict criteria to ensure reliability and consistency. First, the source is very important. It should come from an approved API batch, meaning it's been officially validated, and it must have a high purity level, ideally over 99%. This high purity helps us maintain accuracy in our analyses. Moving on to quality requirements, the material needs to be fully characterized. That means we should understand its exact properties and composition. It should also have a low impurity level, ensuring that impurities don't interfere with our results. Additionally, the compound must be stable over time, so it remains unchanged during storage and testing. And finally, it should be representative of the product we're working with, so it accurately reflects what we're analyzing. In terms of documentation, we require a certificate of analysis, which certifies the material's properties and purity. The batch manufacturing record is also essential, providing detailed manufacturing data and stability data that confirms the material's long-term stability. Altogether, these criteria ensure that our working standards are dependable, traceable, and suitable for precise analytical work, which is fundamental to maintaining quality and consistency throughout our process. Let's walk through the procedure for preparing a working standard, which is a critical part of quality control in the lab. First, we start with the selection of a candidate material. This is the raw substance that we believe has the right properties to serve as our standard. It's essential to choose the best quality material to ensure accurate results later on. Next, we move on to sampling under controlled conditions. This means collecting a representative sample while maintaining strict controls to avoid contamination or bias. Proper sampling is vital because it directly impacts the reliability of our standard. After sampling, we verify the identity of the material. We need to confirm that what we have is indeed what we think it is, using appropriate analytical techniques. This step helps us avoid any mix-ups that could compromise the entire process. Then, we qualify the candidate against a reference standard. Essentially, we compare it to an already established standard to ensure consistency and accuracy. This step helps us validate that our candidate material meets the necessary specifications. Once qualified, we determine the potency or perform an assay. This is where we measure the exact concentration or activity of the substance, ensuring it has the correct strength for use as a standard. Following this, we prepare the working standard stock. This involves creating a concentrated solution or stock that can be accurately diluted later for routine testing. Proper preparation ensures reproducibility and precision in subsequent analyses. Labeling and documentation are next. It's crucial to clearly label our stock with all relevant information and keep detailed records. This ensures traceability and consistency over time. Finally, we store the working standard under specified conditions, such as controlled temperature or humidity, to preserve its stability. Proper storage maintains the integrity of the standard throughout its usable life. Overall, this systematic approach ensures that our working standards are reliable, accurate, and consistent, fundamental principles that underpin high-quality pharmaceutical testing and compliance. Let's talk about the qualification of the working standard. Essentially, this involves comparing our testing standard to a pharmacopoeial reference standard. This comparison ensures that our standard is reliable and consistent with recognized benchmarks. When we perform qualification, there are several key tests we typically run. These include identification tests, which can be done using IR, UV, or HPLC methods to confirm the substance's identity. We also determine the assay, which tells us the concentration or potency of the active ingredient. Additionally, we look at the impurity profile to identify any unwanted substances, measure the water content to ensure proper stability, and check for residual solvents that might be present from manufacturing. Now, the most critical aspect is the acceptance criteria. Results from our tests must be equivalent to those of the reference standard within specific limits. This ensures that our working standard maintains its integrity and suitability for quality control. By thoroughly qualifying our working standards, we uphold the overall quality and safety of our pharmaceutical products, which is at the core of our entire quality assurance process. Now, let's move on to the next steps involved in establishing a working standard for paracetamol. First, in step 4, we calculate the potency of our sample relative to the USP reference standard. This step is crucial because it tells us how the sample compares in strength to the official standard, ensuring consistency and accuracy. Next, in step 5, we assign a specific potency value, such as 99.7%, based on our calculations. This assigned value provides a clear, quantifiable measure of the sample's strength. Finally, in step 6, we label the sample as a paracetamol working standard. This label indicates that it's a validated, reliable standard for use in quality control and testing. To give an example, when preparing an API working standard for paracetamol, we start by obtaining the USP paracetamol reference standard. Then, we select a high-purity batch of the API, ensuring the quality is as close to the ideal as possible. After that, we perform qualification tests, including HPLC assay, IR identification, and related substances testing, to confirm the sample's identity and purity. By following these steps, we ensure that our working standard is accurate, reliable, and compliant with pharmacopoeial requirements, supporting consistent quality in pharmaceutical manufacturing. Let's look at an example of finished product testing, specifically focusing on paracetamol tablets. In this case, we're performing an assay to determine the exact amount of active ingredient. We start by using a working standard in our HPLC assay method, which helps us establish a reference point for comparison. The procedure involves preparing a standard solution from the working standard and a sample solution from the tablets themselves. These are then injected into the HPLC instrument. The key here is to compare the peak areas generated by the sample and the standard. By doing this, we can calculate the assay using the formula shown. The ratio of the sample peak area to the standard peak area, multiplied by the ratio of the standard weight to the sample weight. This process ensures accurate quantification, which is crucial for quality control and ensuring the product meets the required specifications. It ties back to our overall theme of validating the quality and safety of pharmaceutical products through precise testing methods. Let's take a closer look at an example label for a working standard, specifically paracetamol WS. On the label, you'll see the batch number, in this case WS para 01, which helps us trace the specific production batch. The potency is listed at 99.7%, indicating the strength of this standard. The date it was prepared is 10th March 2026, and it has a requalification date of 10th March 2027, ensuring the standard remains valid over time. Storage conditions are clearly specified as 2 to 8 degrees Celsius, which is crucial to maintain its stability and integrity. Now, when it comes to labeling each working standard container, there are key pieces of information that must be included. First, the material name, so everyone knows exactly what the standard is. The batch number, as I mentioned, helps with tracking. The assigned potency informs us of its strength. The date of preparation and expiry or requalification date ensure we're using a valid standard at all times. Storage conditions are also important, so the standard is kept in the right environment. Lastly, the label must be approved by an analyst or QC, confirming that it has been reviewed and is fit for use. All these details are essential for maintaining quality, traceability, and compliance in our processes. Proper labeling ensures that everyone handling the working standard has the critical information they need to use it correctly and confidently. Let's talk about storage conditions for working standards. It's crucial that these standards are kept under controlled conditions to maintain their integrity and reliability. Typically, we store most standards in a refrigerator at 2 to 8 degrees Celsius. This helps prevent degradation over time. For more sensitive compounds, storing at degrees Celsius is advisable, as it offers additional protection against deterioration. For hygroscopic materials, which readily absorb moisture from the air, using a desiccator is essential to keep them dry and stable. Now, there are some key requirements to keep in mind. First, protection from moisture is vital, especially for hygroscopic substances. Second, protecting samples from light is important because exposure can sometimes cause chemical changes. Lastly, limiting vial opening is a simple but effective practice. Every time you open a vial, you risk contamination and exposure to environmental factors. So, minimizing that exposure helps preserve the quality of your working standards. Overall, these storage practices are central to ensuring your standards remain accurate and reliable for your analyses and experiments. Now, let's talk about the requalification of working standards. It's important to remember that working standards are not set in stone. They need to be periodically requalified to ensure ongoing reliability. There are several triggers that indicate when requalification is necessary. For example, when the expiry date on the standard has been reached, or if there's a new reference standard lot that we need to compare against. Other reasons include suspected degradation of the standard, which can compromise its integrity, or regulatory audit requirements that demand reassessment to stay compliant. When we perform requalification tests, there are a few key assessments we focus on. These include the assay, which confirms the potency or concentration. Then, we check the identity to verify it's the correct substance. And finally, we perform impurity checks to ensure no unwanted contaminants are present. All of these steps are crucial in maintaining the accuracy and integrity of our work standards, ultimately supporting the quality and safety of our products. Let's wrap up with some key takeaways. Generating in-house working standards is truly essential for pharmaceutical QC laboratories. It helps ensure that our analytical testing is reliable, which is the backbone of quality assurance. Plus, by developing our own standards, we can significantly reduce the costs associated with purchasing official standards. This not only saves money, but also supports our routine quality control processes, keeping everything running smoothly. Maintaining regulatory compliance is another critical benefit. To achieve this, we need to follow some key principles. First, our standards must be traceable back to a reference standard, ensuring consistency and accuracy. Proper qualification of our standards is also vital, along with controlled storage conditions to prevent degradation. And of course, periodic requalification is necessary to confirm that standards remain valid over time. All of these practices together help us produce accurate and reproducible results when testing active pharmaceutical ingredients and finished products. This consistency is what allows us to uphold the highest quality standards in our work. For further support, please don't hesitate to reach out. We're here to help you with any questions or concerns you might have after today's presentation. Remember, the goal is to ensure you feel confident and well-equipped moving forward. If you need additional resources or personalized assistance, just contact us through the provided channels. Thank you so much for your time and attention today. I truly appreciate your engagement and interest. And again, thank you for being part of this journey.